Hello everybody, here is your study guide for our mitosis test. Let's go over vocab first. First word is chromatin. You can remember that because it's thin. Also it ends in IN, just like interphase. Chromatid is the next word. I think of it as a kid. It's a smaller version of a chromosome, so it's just 1/2 of a chromosome. A chromosome is 2 complete chromatids joined in the middle. It's going to look kind of like an X. A centromere is that button that joins them in the middle. A centriole, think about the north and south pole. They're at opposite ends of the cell, and the spindle fibers come out of those. Sister chromatids are identical. Think about sisters looking alike. So this would be both halves of a chromosome, identical copies that you would get from mom or dad. The next word on our list, sorry, it got cut off a little bit here on the video. The next word on our list is tetrad. This is 4 complete copies, 4 chromatids, 2 from mom, 2 from dad. This is present during meiosis 1. Diploid refers to the numbers of chromosome. It means that we have 2 copies of every chromosome. That's the number that we find in body cells. Haploid refers to 1 copy of every chromosome. That's the number that we find in sex cells. Stages of the cell cycle are pictured here. M stands for mitosis. This is the stage of a cell's life that is actually dividing. G1 is a gap phase. It's where a cell is just going to live life and do its thing. S stands for synthesis. A cell is going to copy its DNA in that phase, and that's what happens at the end of interphase, so that we double our chromosomes. G2 is the second growth phase. We're just trying to get that cell big enough that when we split it during the M phase, that there's enough stuff to go in both halves of the cell. As far as those cell checkpoints go, the M phase checkpoint is there to make sure that the number of chromosomes is correct and okay and that everything divided equally. G1 is that little cell healthy enough that we would actually want to copy it. The S phase, we're going to check the DNA and see if there's been any mutations or errors with the DNA. And G2, did it grow enough to split up during actual mitosis? If it fails any of those checkpoints, we have apoptosis occur, and that is supposed to prevent cancer. Cancer cells, of course, are going to pass those even though they shouldn't have and create problematic tumor cells. Apoptosis is programmed cell death. Metastasis, you can remember, M is for moving. These are tumors that break off and spread to a new area. Angiogenesis, that word genesis means creation, so this is the creation of new blood vessels, arteries, etc., that are going to feed a tumor instead of the organs it's supposed to be feeding. Stages of mitosis in order: interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. During interphase, we have DNA in the form of chromatin. Prophase and metaphase, we have chromosomes. Anaphase, those chromosomes get split in half, and we now have chromatids. And then telophase, that chromatid is starting to relax and become chromatin again by the time we reach cytokinesis. Let's review then the advantages and disadvantages of reproducing with mitosis. An advantage is that you don't have to find a mate. It is quick and it is simple. A negative is that we have no diversity. This occurs during bacterial division, and it doesn't allow bacteria to do a lot of evolving and changing because there's no genetic difference between them. They're all copies, clones of each other. So as far as stem cells go, a stem cell can be taken from either an embryo or from an adult donor cell and reprogrammed to become any kind of cell. So they are great because we think we can use them to cure different diseases or repair injuries, things like spinal cord injuries, diabetes, cancer, etc. But the negatives about stem cells is that they are still very experimental. We haven't had a lot of time to work with them, and some people have major moral and ethical concerns as far as the sources and use of those stem cells. All right, really fast, here come the phases of meiosis: interphase, prophase, metaphase, anaphase, telophase, and cytokinesis 1 for meiosis 1, then prophase, metaphase, anaphase, telophase, and cytokinesis 2 for meiosis 2. So why do we have to go through all this process? Why do we have 2 phases of meiosis? The whole purpose is to cut the number of chromosomes in half. So I'm about to show you how that works. We start off at the end with what we know we need, a sperm cell or an egg cell that has 23 chromosomes in it, because we know that sperm plus egg has to equal baby with 46 chromosomes. Well, where did each of those come from? You simply add them together, the 4 split cells at the end. Where did they come from? And then we do the same for meiosis 1. Where did they come from? 46 at each side gives us 92. Now, way back at interphase 1, we know that that normal diploid cell started with 46 chromosomes, but during the S phase of the cell cycle, it doubled. That's how we got to our 92. And that's how we can go from a cell that had 92 chromosomes down to 4 daughter cells that each have 23. Unique thing that happens during prophase and metaphase 1 is that we have this crossing over where dad and mom switch pieces of their chromosomes around to give us some genetic diversity. So at mitosis, we have 2 genetically identical body cells made. In meiosis, we have 4 non-identical sex. All right, this next part gets a little complicated. We know we need to make sex cells, sperm or egg cells, at the very end of telophase and cytokinesis 2 that have half the normal number of chromosomes. So if you're given the fact that a normal sex cell has 23 chromosomes, then from there you can track back and look at the phases of mitosis to figure that out. You know that in their normal diploid cells then you would have 46. Now after interphase that number doubles, so we would get 92. And then after the first division of meiosis 1, what we would have is 46 again, which we divide in half to get the 23. So you can follow that pattern to fill out the rest of the table. Meiosis sounds super complicated, so why would we bother doing it? An advantage is that we get some genetic diversity. Negatives would be that you have to go find a mate and that it's really complicated and you can often have errors in that process. Next up, karyotypes. So what we would do when we scan a karyotype is look for 2 copies of every single chromosome. When we find that, we know that it is a typical or normal healthy chromosome map. Then you would look at the sex cells, the X's and the Y's. If it has 2 X's, it's a female. If it has an X and a Y, it's a male. What we notice here on the second karyotype is, yes, they have 2 copies of everything, but it looks like they're missing a sex cell. So we would call that a non-disjunction disorder or a chromosome disorder, and we would call that a monosomy. So once again, 2 X chromosomes indicates a female. A typical male would be XY, but in general, a Y chromosome makes a male. If you have an extra chromosome for a total of 3 of that type of chromosome, you would have a trisomy. Famous example is trisomy 21 or Down syndrome. And then if you have only 1 copy, we call that a monosomy. And a famous example of that would be Turner syndrome, which is X and no second sex chromosome. All of these are caused through non-disjunction, and non-disjunction is what happens when we have chromosomes that are not equally divided either through meiosis 1 or meiosis 2. Meiosis 1 is the more serious of the 2 because it causes all of the cells to be affected. A non-disjunction during meiosis 2 would just affect some of the cells having either an extra or not having enough copies of a chromosome. Good luck to everybody on the test. Hope you have enough to review with your notes, and I will see you tomorrow.